Hello everyone, and welcome back to another discussion in microbiology. In chapter 25, we're going to be talking about our Rickettsias and Chlamydias, spirochetes, and Vibrios. So these are going to be microorganisms that cause some very common diseases that you may have heard of, and some of them not so much. The chapter is going to go over a whole plethora of different diseases, but we're only going to focus on those that are listed here on this learning objectives page. So if it seems like we're skipping around the chapter, it's because we kind of are, because we're not going to cover every single thing in the chapter. We're just really hitting those hot spots that, for my students, I'm really concerned that they understand. So some hallmarks of Rickettsias. They are very, very small. They're much smaller than what we're used to seeing. So for those gram-negatives and for those gram-positives, for chapters 19 and 20, these are going to be organisms that are much, much smaller. They're also what we consider non-motile, which means they don't have any flagella to move. They're aerobic, and because of their small nature, they're usually intracellular parasites that will actually live in the cytoplasm of the host cell. So they have an outer membrane that's made out of this lipopolysaccharide, and it has endotoxin activity that's associated with it, which could allow for some of their toxicity. They are also unable to withstand and to be able to survive outside of the host, and it can die very quickly. Therefore, they're going to require some sort of vector for transmission. And as I said before, they're obviously intracellular parasites, and they kind of use the host machinery to make ATP and to reproduce. So there are four big genera of diseases in the Rickettsias: Rickettsias, Orientinia, Escherichia, and Anaplasma. So the first one we're going to talk about are the Rickettsias. As I said before, they're non-motile aerobic bacteria housed to their obligate intracellular parasites. They're usually transmitted via arthropod vectors, which means like some sort of insect vector, and the bacteria lives inside the cytoplasm of the cell. So the ones that cause the three species that cause most human infections are Rickettsias, which causes Rocky Mountain spotted fever. We also have Rickettsias parowski, and then we have Rickettsias typhus. So Rocky Mountain spotted fever is Rickettsias. This disease is the most severe, and it's also the most common of the Rickettsias. It's transmitted by ticks. You're probably very familiar with another disease that's transmitted by ticks, and that's Lyme disease. Usually, but unlike Lyme disease, we don't have that bullseye rash that we're usually associated with it. What we actually will see is that the infected individuals have a rash on their trunk and on their legs, and if not treated, then about 5% of patients will die. Now, Rickettsias can be treated with antibiotics. It's totally treatable with that. And the prevention is kind of the same prevention that we have for Lyme disease, and that would be to avoid ticks. So avoiding ticks is the best prevention. And the medication that we use for this antibiotic is doxycycline for adults, and use a different medication for children. Now they do have and for pregnant women. They have a vaccine, but it's they actually don't have an effective vaccine. So when you're going hiking and things like that in areas that are full of these ticks that could be carrying this disease, this vector, then you want to make sure that you have tight-fitting clothing, tick repellent, make sure you remove ticks promptly after you've been around them, and this is especially important in the spring and summer months. So for Missouri, where we live, there actually are quite a few cases that we see of Rocky Mountain spotted fever in this area. So in Missouri, Arkansas, this sort of Missouri Valley, we do see cases of this. So here's the disease and death. We're talking about Rocky Mountain spotted fever. So the first thing happens is that the tick that's been infected with this, it has the Rickettsias in its saliva. And this occurs only after the tick has been fed for at least 6 hours. The active bacteria are released into the host circulatory system as it bites the host. Once the Rickettsias gets into the body, it's going to trigger the host cells to undergo endocytosis, which actually will take in the Rickettsias, and the cells that we're most that are going to come in first contact with it are going to be your blood vessels or the endothelium. So once it's taken into the cell, it lyses the endosome's membrane and it's able to escape the cytosol and go ahead and escape into the cytosol and reproduce in there. They're going to divide every 8 to 12 hours, so they have a medium length generation time. The daughter cells, the daughter Rickettsias escape from the cytoplasm, and then they go affect other endothelial cells. So the new generation is going to go on to affect another group of cells. So the Rickettsias doesn't really secrete any toxins, and disease is not a product of the immune response, but what happens is that there's damage to those endothelial cells that leads to the blood cells, the red blood cells leaking into other tissues, and that's going to cause a drastic drop in blood pressure and can cause the patient to go into shock. And then also you have ineffective nutrients and oxygen supplies to other tissues as a result of not being able to have adequate blood flow. So there's a picture, an actual picture from a transmission electron microscope of the Rickettsias that are escaping the endothelial cells. So some of these signs and symptoms, after about a week of infection, patients are going to have fever, headaches, chills, nausea, vomiting, and in a good chunk of cases, maybe like 80 to 90% of the cases, you're going to see the patients have this spotted rash, so that's why it's got the spotted fever, or Rocky Mountain spotted part of it. They have this kind of spotted rash, and it's going to be on all of their trunk and appendages, and even their palms of their hands and soles of their feet, which is usually not the site for rashes, like chicken pox. It kind of looks like chicken pox, but or measles, but the fact that it's on the palms of the hands is a little bit different. In about half of patients, the rash develops into subcutaneous hemorrhaging, and in severe cases, the respiratory and central nervous system and even the gastrointestinal system start to fail. In that case, even with treatment, a good 5% of patients can die. So our next Rickettsias Berlotsky is what we causes epidemic typhus, which is also called louse-borne typhus. So a louse is another arthropod that's called body lice. So body lice can transmit this bacteria to humans. In this particular type of Rickettsias that causes epidemic typhus, the hosts are the primary, or the humans are the primary host. We see it typically happening in overcrowded areas with very unsanitary conditions. So think refugee camps. We see this very prominent because we have large groups of people that are living in a very small area. Outbreaks and we usually see this in more impoverished places, so like the places that are listed, places in Africa, Central and South America, and China. We usually see more outbreaks that are happening in the winter when it's more difficult to wash your clothing every day and also to wash your bedding to get rid of the lice. Epidemic typhus in humans will show itself with a high fever, mental and physical depression. There's a rash that lasts about 2 weeks, and in really, really bad cases, especially from those that are very old, it can actually lead to blood loss to the arms and the legs, and can lead to the loss of digits because there's insufficient blood supply. Deaths from epidemic typhus results in about 60% of people that are untreated, and they're usually older patients, and then only about 20% of younger, healthier patients will see death. The way that they're able to diagnose it is by observing for signs and looking for pictures of lice, that not pictures, but the signs and symptoms of lice. Typhus can reoccur many years and decades after the first episode. We can see it coming back. Now, the way we treat it is just with doxycycline, the same thing that we treated Rocky Mountain spotted fever with. Prevention is just good personal hygiene. And we do have a vaccine available for very huge, it's an attenuated vaccine for a very high-risk population. So here are a list of some of the Rickettsias, and the ones that we've talked about are the ones that I'm most concerned with you being able to discuss on your exam. So I'm just going to give you all a second to just kind of go over this chart. Alrighty, so now on to chlamydias. Now, chlamydias used to be considered a virus. There are a group of bacteria that kind of vie, or they're kind of in competition with Rickettsias of being the smallest of bacteria, because Rickettsias are really, really tiny. Remember, there's these obligate intracellular parasites the Rickettsias are, and they're just super, super small. Like Rickettsias, chlamydias are non-motile. They don't move. They grow and multiply within vesicles in the host cells. And we used to think they were viruses because they were so small and they had this kind of virus lifestyle. They're obligate intracellular pathogens on here. They do have cellular aspects to them, and they are cellular. They've got genetic material. They have functional 70S ribosomes, and they do have two membranes, kind of like a gram-negative bacterium, but they don't really have that peptidoglycan between the membranes. So in that sense, chlamydia is really just kind of lack cell walls. They have an outer membrane, but they really don't have that cell wall. Unlike Rickettsias, chlamydias do not have anthropods or vectors or hosts. So they have some very different or more unusual features. So as a result of that, they have their own phylum, Chlamydiidae. They have a very unique developmental cycle that involves two forms. You have the tiny cocci called elementary bodies, and then you have the larger pleomorphic reticular bodies. Elementary bodies are usually dormant and resistant to environmental extremes, so they kind of act a little bit like an endospore. They can survive outside the host cells, and they're just kind of dormant, kind of like an endospore. We don't call them endospores, though. And then we have the reticulate bodies that are non-infective, obligate, and intracellular that can replicate via binary fission. So with the smaller bodies, they're more infectious. The elementary bodies are infectious, the reticular bodies not so much. But both forms can happen in the phagosome, which is the phagocytotic cell, the cell that pulls in the bacteria, puts it into its little vesicle, and fuses with a lysosome. We call that a phagosome. So they can both happen within the phagosome of a host cell. So here we have the life cycle of chlamydia. So once an elementary body attaches to the host cell, it's then going to trigger endocytosis into the host cell. Once inside the endosome, the elementary body converts it into a reticular body. That reticular body is the one that can divide very rapidly and give multiple reticular bodies. Once the infected vessel becomes filled with these reticular bodies, it's called an inclusion body. And about 20 hours after infection, the reticular bodies within the inclusion body convert back to elementary bodies. And then a few hours after that, the elementary bodies are released into the host cell via exocytosis, and then they can affect a new cell and complete the life cycle. So the first chlamydia that we're going to talk about is Chlamydia trachomatis. So Chlamydia trachomatis has a very limited host range, and that very limited range consists of humans, except there's one strain that is a little bit different. It enters the body through cuts and the skin, so through abrasions and lacerations, and infects a whole host of different types of cells. But even though it can affect a whole host of different types of cells, it does have, like I said before, a limited range of what strains can affect humans. So it can affect the conjunctiva of the eye and mucous membranes. Chlamydia is the most commonly reported, reportable sexually transmitted disease. Clinical manifestation of the disease are cell destruction and inflammation. It also causes it's a leading cause of blindness worldwide that is actually preventable or treatable. Usually in very poor, crowded communities where there's inadequate personal hygiene and adequate sanitation, we see this bacteria causing blindness for people. So Remember we said that it can infect the conjunctiva and the mucous membranes of the caruncle of the eye. So diseases that are caused by the various strains of Chlamydia trichomatis, there are two main types. We have the sexually transmitted diseases and the ocular disease, which we call trachoma. Trachoma is the one that causes blindness. So the sexually transmitted disease, so-called the LGV strain of Chlamydia trichomatis, causes what we call lymphogranuloma viridium. Actually, what we start to see are that you initially have genital lesions around the genitals that are the forms of these buboes. The buboes get really, really big, and then they rupture, and they have these kind of draining sores that come around of it. A lot of times, and we can find these things usually near the site of the initial infection, on the penis, urethra, scrotum, in females, the vulva, the vagina, the cervix, or in the rectum. The lesion is usually overlooked because it's small, it's kind of painless, and it usually heals very quickly. However, there are other signs and symptoms that we see with this. Headaches, muscle pain, and the fever can happen with this very early stage of disease. The second stage of the disease is these much larger buboes, or these swollen lymph nodes that are associated with the lymphatic vessels. At this point, the buboes can start to drain, and they are accompanied by fevers, chills, anorexia, muscle pain, just very uncomfortable. A lot of these infections in women are asymptomatic, like 80%. More than 75% of men have symptoms, and infected men, it can actually lead to urethritis, which is inflamed urethra, which can be distinguished from gonorrhea, because some people think that because it hurts to pee, if they're infected with chlamydia, that it is gonorrhea. So some of that can be difficult when it's chlamydia urethritis. And this accounts for about half of non-gonorrheal urethritis, inflammation of it. And pelvic inflammatory disease reinfections can cause pelvic inflammatory disease. Proctitis, it's actually from the Greek word, which means rectum, and itis means inflammation. Can happen in men and women as a result of the bacteria from the vagina, the vulva, the cervix, the urethrum to the rectum. And about the smaller chunk of these cases, maybe 15 to 20% of proctitis happens in homosexuals, and as a result of spreading the Chlamydia trichomatis as a result of anal intercourse. And then what we're looking at here is an advanced case of a lymphogranuloma. It's a large bubo, and you can see kind of its crusting and where it's healed a little bit, but this is definitely a spot where there's been some drainage of fluid, and where there's that drainage of fluid, there's definitely going to be the ability to pass that infection on to someone else, just from contact alone, if there are any abrasions or cuts or anything like that. So the next type of the class of disease that we go in here from that are caused by the same strain of Chlamydia trichomatis is trachoma, which is the ocular disease. And as I said before, it is the leading cause of non-traumatic treatable blindness in humans. What's happened here is that the bacteria are actually scarring the conjunctiva, which causes the eyelashes to kind of turn inward. So instead of the eyelashes turning out, you know, your pretty Betty Boop eyelashes turning out, the conjunctiva is inflected and inflamed, and so the eyelashes are going to turn in. And as they turn in, they actually will scratch the cornea, that clear outer surfacing of the eye. So as it's scratching that cornea, it can lead to blindness. So we typically see these infections occurring during childbirth, where mother passes on the infection to her child, and as a result of that, it kind of scratched. You know, the child's going to be what we consider non-traumatic blindness. I forgot to discuss this with the trichomatis. The way that we prevent this is by using abstinence and faithful monogamy, and also condoms help to provide protection. But we do have treatment for it in the form of antibiotics, and then to prevent chlamydia, trichomatis, the ocular disease, we do put those eye drops in, that same eye drops that we put in for Neisseria gonorrhea, to put those eye drops in the baby as they pass through the birth canal as a prevention. So here is a picture of that inflamed conjunctiva. So the underside of the eyelid is affected with a trichomata on there, and those eyelashes, they'll actually start to turn inward and grow inward as opposed to growing outward because of this swollen area and all of the infection, the inflammation here. It's going to actually cause the eyelashes to turn inward and scratch the cornea. So diagnosis, prevention, and treatment. Diagnosis, we have to actually see the bacteria inside the cells from that. And if we do see that, we prescribe doxycycline and erythromycin to eliminate the genital infections. And we could give those same antibiotics for ocular infections. We can correct the deformities that happen from trachoma, the ocular disease, to prevent blindness. And then prevention is abstinence to prevent sexually transmitted infections, or the use of condoms, responsible use of condoms. And then blindness is prevented by the prompt use of antibacterial agents once the infection has been identified, and quickly identifying the infection. Chlamydia pneumoniae causes bronchitis, pneumonia, and sinusitis. Most infections only produce just like a crappy feeling. You kind of just feel malaise and just kind of feel a little icky. Some cases, however, are characterized by a development of severe pneumonia, and sometimes you can't even distinguish that from what we call primary typical pneumonia, which is caused by a different organism, Mycoplasma pneumoniae. We treat it with doxycycline and erythromycin, and even with those treatments, some infections are going to persist. Prevention is difficult because Chlamydia pneumoniae is pretty ubiquitous. So the bacteria is spread via respiratory droplets and kind of all over. And here we have a comparison of some of these smallest microbes between Rickettsias, Chlamydias, Mycoplasmas, and viruses. So Rickettsias and Chlamydias and Mycoplasmas are all cellular, and then viruses are the only ones that are acellular. So we kind of have a look at that. So this whole entire time I was talking, you all weren't hearing anything. So primary syphilis typically is going to affect, that's the initial infection of it. You have these cankers that we associated with it. Secondary syphilis, we see a rash associated with it. Typically, in either primary or secondary is the time when someone's going to get antibiotic treatment for it. Latent syphilis means that the rash has gone away, and you can be in a latent stage for like 30 years. But a good chunk of those people that have latent syphilis, they're going to it's going to become tertiary syphilis. Tertiary syphilis is what Al Capone had, and Al Capone syphilis, at the tertiary stage, you don't really see the issues being from the bacterium itself. What you actually find is the problem is that there has been hyper immune response as a result of the continuous infection for so long. So you'll have a drop in the function of various different organ systems, including dementia. So for Al Capone, he got really lovey-dovey and was really nice when he was in prison, and he got his late stages of syphilis because he just kind of had this dementia. Congenital syphilis is syphilis that the mother has and passes it on to her unborn child. And as she passes the child via the placenta, if it happens in if it's primary or secondary, the mother's in primary or secondary stage of syphilis and is not getting treatment, then that can result in the fetus dying. If it is a latent stage or in the tertiary stage of it, then the child can have, the fetus may live, but it can result in mental retardation and malformation of various different fetal organs. So in this picture here, we're looking at those cankers that we associate with primary syphilis, the widespread rash over the body and secondary syphilis, and then this guma, which is this kind of rubbery, sort of very painful, and that looks actually to be quite painful, lesion that we see on the skin or the bones of tertiary syphilis. So diagnosis, we use antibody tests against bacterial antigens. So we use fluorescence tests to kind of coat the suspected fluid from the person, from the patient, with these fluorescent antibodies and look for them to glow under the microscope. So we use fluorescent microscopy as a diagnostic aspect of that. Penicillin is usually the treatment of choice for either primary, secondary, latent, and congenital syphilis. It's not really that infectious for tertiary syphilis because at this stage the immune system has a hyper response to it. It's not really an active infection. Patients try to desensitize patients, or physicians try to desensitize patients who have penicillin allergies. Tertiary syphilis treatment is really difficult to diagnose because it mimics so many other different diseases. Very few of them have those spirochetes present. Prevention is abstinence and to practice safe sex. And definitely to, for any of these diseases that we talked about, whether it's chlamydia or syphilis, or rickettsias, is to seek the treatment of a physician in order to get antibiotics, because these diseases that we have talked about today are all completely treatable. So we have non-venereal treponema diseases. So treponema pallidum was the syphilis one, and that is what we call venereal disease. So non-venereal diseases, we usually see them in impoverished children in unsanitary conditions that we'll find these diseases. We can use penicillin to treat them, and prevention is by limiting contact with these infected skin lesions. And this is just a patient with yaws that has the draining lesions that are characterized by the later stages of the disease. On to Borrelias. Members of the genus Borrelia are lightly staining gram-negative spirochetes. They're larger than species of Triponium. They're usually gram-negative bacteria that lack that outer membrane. Instead, they have this really unique glycolipid layer around them. Species of Borrelia cause two main diseases, Lyme disease and relapsing fever. So Lyme disease, caused by Borrelia burgdorferi, is a causative agent. Borrelia burgdorferi is a causative agent of Lyme disease. Lacks the iron-containing enzymes and proteins. Bacteria are transmitted via humans to humans via a tick bite. And what was something else that we talked about In this lecture alone, that was transmitted via a tick bite. Rocky Mountain spotted fever, right? The tick life cycle is very important in understanding how Lyme disease is spread. So for that life cycle, I thought I had this. Okay, we do have slides. So here we have the red blood cells, and we have the spirochetes, the Borrelia or Burgdorfii. So first thing that happens here, so the first thing that happens is that the transovariant transmission of Borrelia is really kind of rare. So the tick's going to hatch in the spring and are uninfected. First, the larvae become infected during their first blood meal. During the first blood meal. Then the remainder of the year, the larvae are going to digest the blood meal while the Borrelia, the bacteria, replicates inside the tick. Then, in the spring of their second year, the ticks will molt into what are called nymphs, and they will feed a second time. The second time, they infect the new host with Borrelia because of their saliva when they bite the new host. The uninfected nymphs then next can happen is they burrow into, the uninfected nymphs can become infected in this time if they feed on an infected host. The next thing that we have is that the male ticks will feed on a deer and mate in the fall of the second year. The female ticks lay unaffected eggs, and then we have the larva, and during this time here, you can also have the tick biting, the infected tick biting a human. So for this life cycle, as I said before, it's really important to kind of have an understanding of how their life cycle works because it's very it's important in how the transmission of the disease moves. So Lyme disease has a broad range of signs and symptoms on here. There are three phases of the disease in untreated patients, and what we find here So what we find here are three diseases in untreated patients. We have that bullseye rash, that's kind of a characteristic, and it doesn't happen in all patients. But when we do find that, that's usually a nice little, yep, this is Lyme disease. We can have neurological symptoms and cardiac dysfunction. Severe arthritis can last for a very long time and actually last for years. And that's a result of the body's own immune system trying to deal with the infection. Increase of cases in the United States because humans are coming in closer association with deer ticks, just the sheer nature of being out in the out in areas where you'd find much closer contact. And here's an incidence of where we usually find Lyme disease. You'll notice that we see it on the rise. So how we treat it and diagnose it, we usually look at those signs and symptoms, and we do have serological tests that we look at to look for that spirochete and identify it. Drugs, antibiotics are effective during the first stage of Lyme disease, but in the later stages of it, sometimes the symptoms can be more difficult to see, and a treatment is also much more difficult. Best prevention, like the same with the Rocky Mountain spotted fever, is to avoid ticks, and also to use repellents such as DEET are actually useful. So relapsing fever is another species of Borrelia, and it can cause two types of relapsing fever. Louse-borne relapsing fever is transmitted between the human and the body lice, again, pediculus humanus. A disease known as tick-borne relapsing fever occurs when any several species of Borrelia are transmitted between humans by soft ticks. So soft ticks are what are transmitting it. We see this primarily in Africa and South Asia, the louse-borne relapsing fever. The tick-borne relapsing fever we see kind of worldwide. Characterizations of theirs are recurring episodes of sepsemia and fever as a result due to the body's repeated efforts to try to get rid of the spirochete. Observation of relapsing fever. Diagnosis, we want to observe the spirochetes as the primary method of diagnosis. And like with Lyme disease in the very first stage, it's actually treatable with antimicrobial drugs. Treatment or preventative measures, avoid ticks and lice, good personal hygiene, and the use of chemical repellents are effective. Leptospira. Leptospira introgens is a motile obligate aerobic bacteria found in wild and domestic animals. Leptospirosis enters the skin or mucous membranes and then travels into the blood system throughout the body. It can cause hemorrhaging of major organs such as your liver and kidney, and as a result will lead to this dysfunction. This occurs pretty much worldwide. It's not just in one place that we see this. We get it with person-to-person spread. We don't really see the transmission from animals to humans. For people that work with animals a lot, there is a vaccine that's available. It's not for people who work with animals, but for the animals themselves, for livestock and for pets, there is a vaccine available. And it is treatable with intravenous penicillin. We can't really get rid of it because there's so many animal reservoirs. And there is leptospira. So far, the 3 spirochetes we talked about has been Treponema or syphilis. We talked about Borrelia, which causes Lyme disease, or relapsing fever, and Leptospira, which is also a spirochete. So our next one that we'll talk about are Vibrios. So Vibrios have many different characteristics of those enteric bacteria that we talked about in chapter 20, and we found Vibrios in water environments worldwide. Vibrios consistently cause a cholera. So this is kind of a recap of where we've been. We've talked about a bunch of different gram-negative bacteria, the Rickettsias, the Chlamydias, and the spirochetes. Now we're going to look to this last group here. So Vibrios, the characteristic of them, they have these really cool curved rods that we see there in the back. They are gram-negative, and they kind of act, like I said, those other enterics, E. coli and salmonella, and they also have that O polysaccharide antigen. So Vibrio is a common pathogenesis, epidemiology of disease. Common species infect humans. The bacteria can live in fresh water. The most common Vibrio, the one that we're going to spend a good chunk of time talking about, is cholera. Humans become infected by ingesting contaminated water and food. We find cases of cholera that are happening as a result of these Vibrio in places that have very poor sewage treatment and water treatment. A characteristic of this Vibrio are what we call these rice stools that we find here. The most recent kind of epidemic of cholera that we've seen has been down in Haiti after the earthquake they had in 2010. So after that earthquake, and even though it's been several years since they've had that earthquake, they've been unable to get their sanitation system up properly. And then there's also a lot of indication, and the United Nations has even agreed to this, that there have been some UN workers that have brought Vibrio to Haiti in their attempts to try to help. So aid workers from Asia in 2020 brought it to Haiti, and it spread now to the Dominican Republic, and we also now in Cuba see it there as well. The most important virulence factor of cholera is the cholera toxin, and as a result of that toxin, it actually can lead to that severe loss of fluid and electrolytes. If not treated, cholera can be it has a high mortality rate. It can be deadly. So here we see some kind of like an initial epidemic that started, and then we watch how it spreads. And then the second wave of epidemics that we have, notice that it's not going to be the same color, because in Haiti, and the Dominican Republic, and now even over in Cuba, this is the most recent wave of it. In Haiti, half of that island is Haiti, half of it is the Dominican Republic. That's from a UN aid worker coming from Asia that brought the disease from Asia there. And then there's poor sewage treatment because of the devastation of the earthquake, that we see the water is infected. So diagnosis is usually based on the actual characteristics of the diarrhea. Rarely is an observer going to actually find a Vibrio cell in the water stool of the patient, but it's usually diagnosed because of the way that the diarrhea looks. So treatment for it is antimicrobial drugs can reduce the exotoxin production and the volume of diarrhea. Fluid and electrolyte replacement is a very important aspect of treatment to replace the fluids that are lost. And then prevention is just proper sewage treatment and water treatment can prevent these epidemics. But in these developing countries and countries that have been just ravaged by either war or have been ravaged by earthquakes, that can actually keep their water treatment facilities from being prepared or developed the way they need to. There is a vaccination that does provide some protection. So the next disease we'll talk about is Campylobacter jejuni. It's likely the most common cause of bacterial gastroenteritis in the U.S. Like Vibrio, it's also gram-negative, slightly curved, and it is motile by means of just one polar flagella like we see for the cholera, Vibrio cholera. Many animals are reservoirs for the bacteria. Humans typically become infected by consuming contaminated food, milk, or water. The most common source of infection is poultry. Usually these bacteria are self-limiting, and they just result in bloody, frequent diarrhea. Even though it's self-limiting, it's shed right out in your stools. It is not comfortable. Treatment for it is prevention, proper food handling, and preparation can reduce the spread of this bacteria. Helicobacter pylori is a slightly helical, highly motile bacterium that usually likes to hang out in the stomach of its host. The way that we recognize it is based on the ribosomal RNA nucleotide sequences, and it has a slightly different classification. Unlike Campylobacter, Helicobacter doesn't reduce nitrates, has flagella, and is urease positive, so there are different biochemical tests that we can use to separate the two. Today, Helicobacter causes gastritis, which, gastro meaning belly in Greek, and itis, inflammation. And what you probably most commonly are used to associating Helicobacter pylori with is ulcers, peptic ulcers, which are just erosions of the mucous membranes of the stomach. We at first didn't think that it was a bacteria that caused ulcers. We thought it was stress, until we actually had a physician who was willing to drink his culture of Helicobacter, and he actually developed ulcers. So as a result of that, he won the Nobel Prize for his sacrifice in 2005. There's lots of virulence factors that will help the bacteria colonize the stomach. It's got proteins that will prevent the production of stomach acid. It can also live in a more acidic environment than most bacteria. It has flagella that actually allow it to burrow through the stomach lining, and because of the shape that it has, it can move through the stomach lining very easily. It has adhesions that will allow it to bind to your gastric cells, and then it also produces enzymes that will help to neutralize stomach acid. So it has all of these different virulence factors that will allow it to the stomach to be an optimal place for it to hang out, cause infection. So the role of Helicobacter is forming ulcers here. They invade the mucus and attach to the epithelial cells of the stomach, or those gastric epithelial cells. Notice it has all of those tufts of flagella hanging out the end of it. Its toxins and inflammation causes the layer of mucus to become very thin. So this is the mucus, and as a result of having all of those different enzymes that neutralize stomach acid and those toxins that will cause inflammation of the cells. The cells don't produce, those gastric cells don't produce stomach acid like they're supposed to, so it kind of degrades away at that. And then the gastric acid, because we don't have that layer of mucous membrane to protect the stomach, that gastric acid is going to erode or destroy the epithelial cells and cause this ulcer. So the presence of Helicobacter pylori can be demonstrated by a positive urease test, which we kind of talked about, and we said already biochemical tests are a key to definitive identification. We can treat this with antimicrobial drugs that will inhibit and drugs that inhibit acid production to reduce the amount of acid so that we don't have those ulcers taking place. And then prevention is good hygiene, adequate sewage treatment, and just proper handling of food. All right, so that is the end of this chapter. So I hope you enjoyed this and you weren't too annoyed or distracted with my small crying children. Have a great morning, noon, or night.